Okay, thank you for the chance that given to me. My name is Tekla Kalalo, and now I will presenting about advanced biopharmaceutics test. And the title of my topic is an mRNA vaccine against SARS-CoV-2. Okay, now I will explain a little bit about what is COVID-19, why we must learn about vaccine, or why this vaccine is need for COVID-19 pandemic. COVID-19 or coronavirus disease 2019 has resulted in nearly 15 million infections, claiming more than 1 million human lives globally. This COVID-19 is caused by severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2, a beta coronavirus that emerged from bats and then transfers to humans through intermediate hosts. SARS-CoV-2 transmits from human to human via respiratory droplets, leading to a respiratory tract infections that can progress severe pneumonia, multiple organ involvement, and fatal outcomes. This outbreak was followed by the release of the first situation report from the World Health Organization or WHO on January 2020, and since then a global pandemic has resulted in an increasing number of deaths. So, a vaccine is urgently needed to control the current exploding global pandemic of COVID-19 and prevent recurrent epidemics. In this picture, we can see structural features of SARS-CoV-2. SARS-CoV-2 is an enveloped single-stranded RNA virus. The viral envelope is embedded with spike or S glycoprotein, the matrix or M protein, and envelope or E protein. Encased into this envelope is a positive single-stranded RNA is a viral genome bound to the helical nucleocapsid and protein. The viral spike protein that mediates entry into the host cell has been identified as one of the preferred vaccine targets. SARS-CoV-2 targets the ACE2 receptor on the host cell via its spike protein S composed of S1 and S2 subunits. The S1 subunit contains the receptor binding domain or RBD that interacts with the ACE2 receptor, thereby inducing a series of conformational change facilitating membrane fusion and entry. Due to the critical role played by the spike protein in mediating viral attainment and entry, nearly all SARS-CoV-2 vaccines currently in development are predominantly focused on eliciting protective immune response targeting the viral spike. mRNA-based vaccines have become a potentially preventive approach as they are safer, more efficient, and easier to manufacture. Like the DNA vaccine, no mRNA vaccine has been approved for human use. An mRNA vaccine, mRNA 1273 for SARS-CoV-2 encoding a provision stabilized form of its spike S protein, has been co-developed by researchers at the National Institute of Allergy and Infectious Diseases or NIAID and at Moderna. This is the first mRNA vaccine to go into clinical trials for the safety and immunogenicity assessment. A large phase 3 efficacy trial evaluating a 100 microgram dose has already begun to further assess the mRNA 1273 vaccine in approximately 30,000 adult volunteers. Alternatively, Pfizer and BioNTech vaccine candidates based on mRNA encoding SARS-CoV-2 are BD complex with the lipid nanoparticles are also under phase 3 study. And other mRNA candidates developed by CureVac and Arcturus or Duke NUS are also rapidly progressing through phase 2 trial for assessment. This is the schematic diagram of the mRNA-based vaccine targeted to the spike protein or S protein of severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. The mRNA-based vaccine targeted to the S protein of SARS-CoV-2 works by active immunization. This technique will not use part of the virus but only recombine mRNA of the S protein in vitro according to gene sequence, which is coated with lipid nanoparticles for effective delivery. Once injected into the muscle, the myocytes take up the lipid nanoparticles and then release the mRNAs into cytoplasm for translation into the S protein. This endogenously synthesized S protein will be secreted to active both humoral and cellular immune response. This is the potential advantages of mRNA-based SARS-CoV-2 vaccines. So, there are four main safety and efficacy advantages of the use of mRNA-based antiviral vaccines over traditional approach. First, mRNA-based antiviral vaccine minimize the potential risk of infection and insertion induced mutagenesis due to natural degradation of mRNA in the cellular microenvironment. Second, the immunogen's high efficacy due to engineered mRNA structural modifications improves its stability and translation efficacy. And the third, the high potency of mRNA-based vaccine capable of generating potent antiviral neutralizing immunoglobulins with only one or two low-dose immunization may induce strong immune response by activating both CD8 plus and CD4 plus T cells. And the fourth, engineered mRNA production facilitates large-scale production of sufficient vaccine doses required to treat mass populations. All these factors make the mRNA vaccine more suitable for a rapid response to the emerging COVID-19 pandemic. The mRNA 1273 vaccine candidate manufactured by Moderna encodes the S2P antigen, consisting of the SARS-CoV-2 glycoprotein with a transmembrane insert and an intact S1 until S2 cleavage site. S2P is stabilized in its perfusion conformation by two consecutive proline substitutions at amino acid position 986 and 987 at the top of the central helix in the S2U subunit. The lipid nanoparticle capsule, composed of 4 lipids, was formulated in a fixed ratio of mRNA and lipid. Okay, for the efficacy and safety of the mRNA 1273 SARS-CoV-2 vaccine, this coronavirus efficacy or COVE phase 3 trial was launched in late July 2020 to assess the safety and efficacy of the mRNA 1273 vaccine in preventing SARS-CoV-2 infection. And for the method that used for the study, this phase 3 randomized observer-blinded placebo-controlled trial was conducted at 99 centers across the United States. Persons at high risk for SARS-CoV-2 infection or its complications were randomly assigned in a 1:1 ratio to receive 2 intramuscular injections of mRNA 1273 100 microgram or placebo 28 days apart. The primary endpoint was prevention of COVID-19 illness with onset at least 14 days after the second injection in participants who had not previously been infected with SARS-CoV-2. Okay, and for the result, the trial enrolled uh, 30,420 volunteers who were randomly assigned in a 1:1 ratio to receive either vaccine or placebo. In this case, 15,210 participants in each group. More than 96% of participants received both injections, and 2.2% had evidence like serologic, virologic, or both of SARS-CoV-2 infections at baseline. And symptomatic COVID-19 illness was confirmed in 185 participants in the placebo groups, in this case 56.5 per 1,000 person years, or 95% confidence interval, or CI, 48.7 to 65.3. And in 11 participants in the mRNA 1273 group, in this case 3.3 per 1,000 person years, 95% CI, 1.7 to 6.0. Vaccine efficacy was 94.1%, and efficacy was similar across key secondary analysis, including assessment 14 days after the first dose, analysis that included in participants who had evidence of SARS-CoV-2 infection at baseline, and analysis in participants 65 years of age or older. Severe COVID-19 occurred in 30 participants with one fatality. All 30 were in the placebo group. Moderate transient reactogenicity after vaccination occurred more frequently in the mRNA 1273 group, so serious adverse events were rare, and the incidence was similar in the two groups. Okay, and for the conclusion, the mRNA 1273 vaccine showed 94.1% efficacy at preventing COVID-19 illness, including severe disease. Aside from transient local and systemic reactions, no safety concerns were identified. And this is the reference that I use for my task. That's all. Thank you.